Hello everyone, my name is Wenyu Chen, and in this video, I'll be talking about some of the neuronal migration disorders known as NMDs that result from mutations in a wide range of proteins in the process of nucleokinesis. First, let's review the proper migration of the neurons. During brain development, radioglial cells, which are the neural progenitor cells, divide in the ventricular zone. They first give rise to intermediate progenitors that will eventually differentiate into neurons, hence neurogenesis. As cells differentiate, they move away from the ventricular and subventricular zones to their destined location. The microtubule cage and the centrosome are responsible for the forward movement of the cells. In addition, renin, a signaling molecule secreted by the Cajal red cell cells, is responsible for binding to the lipoprotein receptors on the migrating neurons and telling them when to stop moving and join the formation of the cortical plates. As you can see, renin allows for this inside-out organization where new neurons travel past the previous neurons to form a new outside layer. It is during this process that mutations in the microtubule-associated proteins and centrosome-associated proteins can lead to human developmental diseases. The first type of NMDs we will discuss is paraventricular nodular heterotopia, which is characterized by possibly no neuronal migration caused by the X-linked dominant mutation in the filament 1 gene. This mutated protein is responsible for cellular structure and the binding necessary for cell adhesion and migration. This mutation is lethal for males, but females with this mutation on one X chromosome will have partial migration. The visualization of this mutation is these disorganized clusters of neurons near the ventricles. Moving on to type 1 lissencephaly, which is characterized by impaired migration. Lissencephaly is often called smooth brain because of the complete absence of gyra and sulci, although the sylvian fissure may still be preserved. This syndrome may result from DCX mutation in males or the deletion of LIS1 gene. Since both are microtubule-associated proteins directly involved in nucleokinesis, their mutation results in defects in radioneuronal migration. Patients with type 1 lissencephaly have severe psychomotor and intellectual impairment, along with intractable seizures. DCX mutation in females, on the other hand, will result in subcortical band heterotopia. This phenotypic difference is again caused by the X-linked dominant mutated gene. Because females have two X chromosomes, they will have partially normal migration and a normal appearing cortex. Instead of impaired migration, type 2 lissencephaly, often called cobblestone lissencephaly, is caused by the over-migration of neurons, which happens when neurons migrate beyond the renin-secreting Cajal red cells in the marginal zone. Similar to type 1, type 2 also results in severe psychomotor impairment and seizures. However, what distinguishes their symptoms is the ocular abnormalities unique to type 2. Most mutations that are associated with type 2 are responsible for glycosylation, which means that the malfunctioning of these proteins will then lead to declined ligand receptor interactions, protein stability, and protein secretion. The brain scans for type 2 patients can be seen with missing cortical layers, disorganized cerebellum, and the thickening of gray matter. And the reason for its name is the visible cobblestone-like lissencephalic cortical surface. The most common NMD is polymicrogyria, which is defined by the excess folding of the cortex that results in way too many gyra-like folds on the brain surface. Although the signs vary widely depending on the severity and the location of mutation, epilepsy, developmental delay, and muscle weakness of either hemiparesis or quadriparesis are some of the possible symptoms of polymicrogyria. Interestingly, one mutation of this syndrome is on the PAX6 gene. These are all the neuronal migration disorders that I will be covering in this video. Currently, there are more than 25 kinds of NMDs observed. It is important to remember that because each syndrome can be caused by mutations in various genes or a combination of mutations, it is extremely difficult to generalize the symptoms and life expectancy because they vary with the specific mutation and the severity of the mutation. Since it is impossible to compensate for missing brain areas, NMDs today are still incurable, but the patients may be able to control their symptoms using medication.